Anyway, now that we've done the overview, let's go into a bit more detail. I'll start by explaining what FASTQ files are. FASTQ files store DNA sequence data along with the quality scores for each nucleotide. The format could theoretically work for amino acid sequences, but personally, I've never seen it used for that before. So in the context of this video series, I'll only talk about FASTQ as a file format for DNA sequences. In FASTQ files, each data entry represents a single sequence of DNA, known as a read, and consists of exactly 4 lines. The first line contains the read ID. The second line contains the ACGT nucleotide sequence. The third line is irrelevant and often left empty, and the fourth line contains the quality scores of the nucleotide sequence. The FASTQ file format doesn't have a way to store additional information like secondary structure or molecular modifications, but that kind of information doesn't exist in the NGS data anyway. Single-end sequencing will generate one FASTQ file per sample per flow cell, while paired-end sequencing produces a pair of FASTQ files per sample per flow cell, although sometimes that data is stored in a single merged FASTQ file. Paired-end sequencing produces results with higher mappability than single-end sequencing, but costs more. Some techniques or analysis methods explicitly need paired-end sequencing in order for it to work. The FASTQ files that can be used for this pipeline can either come directly from a sequencing experiment, from an online data repository, or both. It's possible to pull together the reads from paired-end and single-end sequencing. However, this can bias the data, so discretionary judgment is advised. Also worth noting is that a lot of bioinformatics software requires paired-end read data to be stored in 2 separate FASTQ files, one for each orientation. Therefore, it is recommended that paired-end data stored in a single file be separated into 2 files, one for each orientation. Okay, so now that we know what FASTQ files are, let's talk about the pre-processing which raw reads undergo before they're ready for alignment. There's adapter and primer trimming, quality control trimming, and the combining or splitting up of files for logistical reasons. These steps can be done in any order because the inputs and outputs are all FASTQ files. As long as the necessary trimming and filtering takes place, the resulting data will be ready for alignment. It should also be noted that some of these steps are not always necessary. Newer sequencing machines usually trim off sequencing artifacts and segregate any multiplexed biological samples into separate FASTQ files, saving us the trouble of having to do it ourselves. Combining multiple FASTQ files into one is only necessary when multiple flow cells were used for the same sample, and splitting up the reads into multiple files is only necessary if the alignment process is being parallelized by multithreading. The alignment process itself is fairly simple. The reads are mapped against the reference genome to see where in the genome these reads originated from. Technically speaking, the reads are aligned against the index of the reference genome. But fundamentally, that doesn't change the fact that we're mapping to see where in the reference genome these reads originated from. The mapping process produces a SAM file. So what is a SAM file? SAM stands for Sequence Alignment Map, and this is the file format used to store alignment results. Some alignment programs produce BAM files instead, but BAM files are just compressed SAM files. They take up less hard disk space on your computer, but they contain identical data organized in the same way as their uncompressed counterparts. I will explain SAM files in detail, but you can also refer to the official SAM file specifications document linked in the description below. The SAM file format can be described as a table with header comments. That is to say, a SAM file consists of 2 parts, a comments section and a data section. The comments section consists of lines which begin with an @ symbol and are typically used to record metadata such as the details of the alignment procedure, which was used to derive the alignment data. The data table contains the alignment data itself in a TSV format. TSV stands for tab separated values. So in raw data form, different rows of the table are separated by new line characters, while the cells in each row are separated by tab characters. Each row of the TSV section corresponds to the alignment results for a single read. The first column contains the read ID. The second column contains information on how the alignment went, and this information is stored as a number which uses the bitflag system. The third and fourth columns contain the chromosomal coordinates of the best mapping result. The fifth column contains the mapping quality score. The sixth column contains more information on how the alignment went in a cigar format. The seventh and eighth columns contain the chromosomal coordinates of the next read in the data. The 10th and 11th columns contain the DNA sequence and quality scores of the read respectively. These are not always identical to that of the corresponding entry in the original FASTQ file. Some SAM files have additional columns with additional information depending on which program generated them. The most important information lies in the third and fourth columns, which contains the chromosomal coordinates of the alignment. Okay, now that we know what SAM files are, we can talk about the standard post-alignment processing steps in the pipeline. These steps are fairly standard across almost all NGS data, because some sequencing and biological artifacts are almost ubiquitous across different NGS techniques and technologies. The steps are as follows. Firstly, if the reads were split up for parallel computing, they have to be recombined into a single file. Then, the alignments are sorted by genomic location. After being sorted, the alignments are deduplicated. Finally, the alignments are filtered by various criteria, such as by the strength of the alignment and by the genomic location which the read aligned to. These steps must be carried out in that specific order. Sorting the reads first makes the deduplication process exponentially faster, so sorting takes place before deduplication. And to catch all duplicates, all reads generated by a biological sample must be examined compared to all other reads, so recombination, when applicable, must take place before sorting. The reason why deduplication should be carried out before filtration is a bit more complicated. It is because during sample prep for NGS, the data is extracted first before being amplified by PCR. Biological noise is present either before extraction or as a result of extraction, and that noise is amplified unevenly by PCR. Therefore, to calculate the true experiment efficacy, the data must first be deduplicated. Of course, doing these steps in a different order will almost never cause a significant difference in the resulting data metrics. Once the experimental noise and general biological noise has been filtered out, the data is now ready to undergo statistical analysis and biological interpretation. This is where the core pipeline ends, because the analyses which need to be performed are going to be experiment specific, and these can be very different from one another. Just to give a few examples, NGS can be used for gene expression analysis. In this scenario, the sequenced DNA is mRNA, and by normalizing the transcripts based on gene size and sample size, we can compare the gene expression profiles of different samples. In contrast, ChIP-seq experiments use NGS to sequence genomic DNA, which was extracted from a pull-down assay, targeting a protein of interest. This allows us to deduce which parts of the genome the target protein is more likely to associate with. Very different. These are just two examples of the many experiments which can be done using NGS technology. But I digress.